Welcome to ANA Investigates, a monthly podcast covering the latest in neurology and neuroscience, brought to you by the American Neurological Association. I'm your host, Adeline Goss. This month, we'll have part 2 of our 2-part series on unbiased diagnostics in neurologic disease. As you may remember from our first episode, UCSF neurohospitalist Megan Richie interviewed Dr. Michael Wilson, a neuro-immunologist at UCSF, about a technique called metagenomic next-generation sequencing, or mNGS. mNGS is a way of searching all of the genetic material in a CSF sample for the presence of any non-human organisms. This approach is thought of as unbiased. It allows us to search for any infectious organism present in the central nervous system, rather than coming up with a list of possible infections and testing for each of them individually. Today, we're back with Megan Richie for part 2 of this series. Welcome, Megan.
Glad to be back.
Megan, when we suspect a patient has autoimmune encephalitis or another autoimmune condition of the central nervous system, the standard approach is to generate a list of known autoantibodies and send the patient's CSF for the tests on that list. Today, you're going to tell us about a different approach, an unbiased technique for the workup of autoimmune central nervous system disease.
Yes, and to learn about this technique, I interviewed my colleague, Dr. Samuel Pleasure, another neuro-immunologist at UCSF. Sam is a part of the UCSF Center for Next-Gen Precision Diagnostics, and he spoke to me about how they developed their technique for novel autoantibody discovery.
What we started to do about 4 years ago was to begin a series of patients using some of the same research cohort that had been being collected by Michael and Dr. Joe DeRisi to try and identify whether some of those patients have autoantibody-associated encephalitis that had been previously unrecognized.
They developed a procedure to see whether any antibodies present in a patient's CSF bind to proteins in the mouse nervous system.
That gives us valuable information indicating that there may be an antibody species in the patient's CSF that is reacting with neural antigens. And then we sort of take that information and go on to do further characterization from there.
So, not to be crude, but they're basically pouring a patient's CSF on a mouse brain and seeing what sticks?
That's pretty much how it works. Anyway, this approach is unbiased because they're not looking for a specifically known autoantibody, but instead looking broadly for any autoantibodies that happen to react to neural tissue, whether they be previously known or unknown.
So what happens once the UCSF team identifies CSF that reacts against neural tissue?
Well, Sam first explained it to me in this way.
When we see an unusual and particularly strong pattern of staining, the evaluation sort of moves on to step 2, which is to take the patient's CSF and use it to probe what we call a programmable phage display library.
So when Sam explained this to me, I realized I really didn't understand what a programmable phage display was and needed some help. It turns out this is a technique that Dr. Michael Wilson actually devised, and so let's go back to hearing his explanation.
So the programmable phage display, this was done together with Dr. Joe DeRisi, also at UCSF in biochemistry and biophysics. And it was expanding on a technique that was published by Steve Elledge's lab at Harvard a few years ago.
Essentially, programmable phage display takes advantage of a library of over 700,000 overlapping human DNA sequences, spanning all the protein coding regions of the human genome.
This library of DNA sequences are then cloned and then packaged into bacteriophage so that the bacteriophage will express the peptide on their surface. And so in solution, you have a library or a population of billions of phage, each of which are expressing one of these 700,000 human peptides.
So when you add the patient's CSF to this mixture, the novel antibody in the patient's CSF binds some of the bacteriophages.
You let the antibodies find their cognate antigen and then do an immunoprecipitation and sequence the region of the phage genome that you know has the human sequence inserted into it. And so you get a readout of the human peptides that were pulled down by the patient antibodies.
So basically, bacteriophages act as a vehicle to serve up all possible human peptides, and then you can see which peptide a patient's CSF sticks to?
Yes, that's exactly it. The research team also has multiple follow-up validation methods to ensure that the peptide they identified is truly the target that produces the staining pattern they observed.
So, but that way, essentially, we end up with first step identifying a patient with putative autoantibodies, and then secondly, moving on to hopefully identify the potential autoantigen, and then thirdly, to validate the association.
So, can you give us an example of how this technique might be used?
Yes, Sam told me an interesting story about one patient from UCSF.
It was a gentleman with progressive cerebellar dysfunction with loss of manual dexterity, which he was able to really notice because of his particular profession required extreme manual dexterity. And he had noticed this as a progressive problem for several years without it being noticeable enough for neurologists to pick up the deficits, actually. He did have evaluations that showed, radiologic evaluations that showed that he had mass lesion that appeared to be a mass lesion in his brainstem, really, upper brainstem. But there also were indications that there were some inflammatory aspects to his MRI and also a progressive cerebellar atrophy. So he bounced around for a bit from UCSF to other institutions, but ultimately his CSF made its way into our research study, where it was apparent that there's a particular staining pattern of his CSF on mouse brain.
So then they ran the programmable phage display and identified an intracellular protein that had never been associated with an autoantibody syndrome in the past.
And with that, that led to testing of a number of other patients who had somewhat similar presentations with cerebellar dysfunction. Turned out that a number of them, 13 in total, were found in this cohort in collaboration with the Mayo Clinic that had autoantibodies to the same syndrome. And they all had as well, they were all male, and they all had germ cell tumors of their testes or dysplasia, dysplastic lesions in the germ cells of their testes. And so this turned out to be what we believe to be a new autoantibody associated paraneoplastic syndrome with cerebellar degeneration that appears to be distinct from the known similar syndromes. And that came directly from this research approach.
Other than identifying brand new autoantibodies, Megan, are there other advantages to this technique?
Yes, and this is where the idea of unbiased testing becomes really important because it doesn't require testing for individual antibodies one at a time.
There may be a spectrum of patients who have more than one autoantibody that can lead to syndromes that are overlap syndromes. We don't know how frequent that is a problem because in the current situation, you have to think of, well, I want to know whether patients with this autoantibody also have antibodies for this. And it's very hard to sort of obviously think of every possible permutation of those combinations.
One question I had was, once an autoantibody is detected, how do we know if it's pathogenic? Sam said there's still work to be done.
What we don't know at this point in many cases is the clinical relevance of the autoantibodies. It's clearly the case that there must be situations where autoantibodies are present in patient CSF that are bystander effects or secondary effects of diffuse inflammation. But these are things that need to be worked out in the future.
That being said, Sam also explained to me that even if an autoantibody isn't pathogenic, it could still serve as a valuable biomarker that indicates an aggressive autoimmune environment. Similar to the way serum neurofilament light chain can be nonspecific but still helpful as a marker of disease progression in some neurologic conditions.
So in what clinical scenario should a clinician be thinking about a novel autoantibody?
Certainly, it could be one possible diagnosis in any patient with an unexplained inflammatory meningoencephalitis. That being said, patients with known primary malignancies are certainly potential good candidates. Another good situation might be a patient presenting with a classic autoantibody syndrome, like cerebellar degeneration or limbic encephalitis.
So it might be useful here to compare and contrast between unbiased testing for CNS infections and unbiased testing for autoantibody syndromes. In both cases, we heard that CSF was the main sample sent for testing. Is there any utility to sending non-CSF tissue for novel autoantibody testing?
I asked that question to both Michael Wilson and Samuel Pleasure. Let's hear from Michael first.
So the test can be done on any tissue type. It's only, but the way that clinical laboratory regulations work is that you need to validate a test for every tissue type you work with. So CSF is the only tissue type that we've validated. We do research-based sequencing on other tissue types, especially brain biopsy tissue, when it's clinically appropriate. It's just that we have to provide a lot of caveats on the results of those tests because it is research and not clinical testing.
That being said, there could be the potential for higher yield of organism nucleic acids when directly sampling infected tissue, particularly in compartmentalized infections such as an abscess. So let's hear from Samuel Pleasure about autoantibody testing on non-CSF samples.
The kinds of tissue we'd be interested in if the patient has a paraneoplastic associated autoantibody syndrome. Where the theoretical basis of that is that the patient's tumor expressed that antigen and presented it to the immune system. So we're certainly interested in getting tissue specimens from patients of tumors that might be associated with their antibody-associated problems, so we could see whether the tumor has the same antigen that we identified as the target of the CSF resident antibody.
I asked both Michael and Sam to describe what they see as the future for each of their unbiased diagnostic techniques, and this is what they answered.
So I think there's a number of exciting directions this type of unbiased testing can go in. One thing that we've been talking about for a while, but is I think really gaining some traction now, is mining the human RNA sequencing data that we get out of every mNGS sample that we run. So I mentioned earlier that about 99% of the data we generate every time we run a sample through mNGS is human gene expression data. And we have some exciting preliminary findings that are not too surprising, that the genes that are up and down regulated in the CSF when a patient is fighting TB meningitis versus when they have cryptococcal meningitis versus when they have anti-NMDA receptor antibody encephalitis, that those gene signatures are quite different based on the type of disease. And so I would love to see that develop as a companion piece of information that is issued with the mNGS diagnosis, being able to say that we did not find an infection in this CSF sample, and the host gene expression signature has a high probability of fitting with an autoimmune encephalitis signature, or we didn't find an infection, but the host gene expression signature looks very similar to what we see in bacterial meningitis.
The other thing is, I think, our screening approach of first using an anatomic screen and then going on to the molecular screens is not at this point clinically validated, but it is in the clinical labs that do clinically validated autoantibody testing. It's something that they do as well, it's my understanding, and they will at times report that information to the clinicians. We've now collected many hundreds of specimens from clinicians at UCSF over the last several years for patients where there is a suspected inflammatory meningoencephalitis. And it is amazing to me the appetite for feedback from those clinicians to try and help them direct therapy. And it is difficult for us to emphasize enough to them that at this point we are not doing a clinically validated test and that the results are not ones that they should be using to direct clinical decision making. However, one could imagine that knowing that a patient's CSF has antibodies present in it that stain mouse tissue, even if the antibodies are not identified as yet and might take longer to identify, could be conceivably valuable information to clinicians. And that's something that we'd like to think about ways to validate.
So, Megan, you've sent both of these tests in your practice as a neurohospitalist. Given what Sam is saying, what do you do when you learn that your patient's CSF contains a novel autoantibody? Do you act on the result?
Well, one thing that both Michael and Sam stressed during our interview was how important it is to understand which tests are clinically validated and which are not. As of now, the only test we have discussed during this two-part episode that is clinically validated is the mNGS testing on cerebrospinal fluid. Unbiased autoantibody testing on any specimen, or mNGS on non-CSF specimens, are research tests only and should not be used to guide clinical decision-making. That being said, if I receive an abnormal result on any of these research-based methods, it can sometimes prompt me to send commercial testing for the potential diagnosis. For example, if a brain biopsy shows nucleic acid belonging to a particular enterovirus, maybe I should work harder in sending validated enterovirus testing to confirm.
Interesting. Well, Megan, thank you so much for teaching us about unbiased diagnostics in neurologic disease.
It was such a pleasure, Adeline. Thank you so much for having me.
Again, that was Megan Richie, a neurohospitalist at UCSF. Thanks also to UCSF neuro-immunologist Samuel Pleasure and UCSF neuro-immunologist Michael Wilson. For ANA Investigates, I'm Adeline Goss.
This has been an episode of ANA Investigates, the official podcast of the American Neurological Association. Thanks for joining us. To learn more about today's program and to access more of the incredible content produced by the ANA, check out myana.org. Adeline Goss is our host. Our producers are Romer Giucaden, Adeline Goss, Michael Halstead, Megan Richie, and Jim Sigler. Our editor is Jen Hurley. If you'd like to learn more about receiving CME credit for listening to ANA Investigates, visit myana.org and navigate to ANA Investigates under the Education tab. For more information not covered in today's program, check out the official summary of this episode, published in one of the ANA's journals, the Annals of Neurology or the Annals of Clinical and Translational Neurology. Copyright, the American Neurological Association.